The human brain is arguably the most complex and magnificent structure in the known universe. A 3-pound universe of electrified jelly containing roughly 86 billion neurons, each capable of forming tens of thousands of synaptic connections, resulting in a neural network so dense and intricate that it outpaces the processing power of our most advanced supercomputers. As we cross the threshold of our 60th year, this miraculous organ undergoes profound and highly documented physiological transformations, shifting from a state of rampant expansion and rapid processing to a phase that requires meticulous maintenance and intentional preservation, demanding a fundamental change in how we manage our daily biological inputs. For decades, the prevailing medical dogma suggested that cognitive decline, the slow erosion of memory, executive function, and linguistic acuity, was an inevitable, inescapable tragedy of the aging process, an insidious fading of the light that we could only hope to delay rather than entirely prevent. However, the dawn of modern neuroscience, propelled by functional magnetic resonance imaging and a deeper understanding of neuroplasticity, has completely shattered this defeatist paradigm, revealing instead that the aging brain remains remarkably malleable, capable of not only halting its own degradation but actively regenerating its structural integrity through a process known as adult neurogenesis. The difference between a sharp, vibrant, and fiercely independent octogenarian and an individual tragically succumbing to the cognitive shadows of dementia does not rely solely on the genetic lottery, but rather is overwhelmingly dictated by epigenetic expression and the daily, seemingly insignificant choices that govern the microenvironment of the central nervous system. We are discovering that the abyss between a thriving mind and cognitive decline can be successfully bridged not by relying on pharmaceutical miracles, but by implementing exact, scientifically substantiated microhabits that cumulatively fortify the neural pathways, clear out devastating neurotoxins, and guarantee long-lasting lucidity and vitality well into our twilight years. To truly understand how to protect your brain after 60, we must dive deeply into the cellular mechanics of aging and explore 5 paramount, seamlessly integrated lifestyle modifications that act as a profound biological armor against memory loss, cognitive slowing, and neurodegeneration. The foundation of this cognitive preservation begins with the absolute optimization of the brain's native waste management network, a recently discovered biological marvel known as the glymphatic system, which operates entirely on the principles of highly specific and structured hydration. For centuries, anatomists were baffled by the brain's apparent lack of lymphatic vessels, the cellular plumbing system that the rest of the body utilizes to clear out metabolic waste, leading to the assumption that the brain simply recycled its own debris or diffused it slowly into the bloodstream. It was not until the 21st century that neuroscientists discovered the glymphatic system, a macroscopic waste clearance pathway heavily dependent on glial cells, specifically astrocytes, which utilize their end feet to pump cerebrospinal fluid through the dense tissue of the brain, essentially washing the neurons and flushing out toxic byproducts. Among these byproducts are beta-amyloid plaques and tau tangles, the highly destructive, misfolded proteins that notoriously aggregate in the brain tissue of Alzheimer's patients, strangling neurons and disrupting synaptic communication until the cells undergo apoptosis or programmed cell death. Crucially, the glymphatic system does not operate at a constant rate. It is largely inactive during our waking hours when the brain is prioritizing cognitive function and sensory processing, and it only truly activates during deep, slow-wave sleep, during which the actual physical volume of the brain cells shrinks by up to 60%, creating wide interstitial spaces for the cerebrospinal fluid to rush through like a restorative biological tsunami. However, this entire magnificent flushing mechanism is fundamentally dependent on the brain's state of hydration, and this is where the first critical microhabit must be aggressively adopted by anyone over the age of 60. Aging naturally diminishes our thirst mechanism, meaning that older adults are frequently in a state of chronic, low-grade dehydration without ever feeling thirsty, which thickens the blood, reduces the volume of cerebrospinal fluid, and utterly paralyzes the glymphatic system, leaving toxic proteins to stagnate and accumulate day after day. Therefore, the simple act of drinking water is elevated from a basic survival necessity to a highly precise neuroprotective intervention, requiring the intentional consumption of water not merely when thirsty, but strategically timed throughout the day, particularly focusing on front-loading hydration in the morning to restore the fluid lost overnight and to optimize blood viscosity for the day's cognitive demands. This microhabit requires waking up and immediately consuming a large glass of water, ideally infused with trace minerals like sodium, potassium, and magnesium, which act as the electrical conductors of the nervous system, facilitating the action potentials that allow neurons to fire and communicate. Proper intracellular hydration ensures that when night falls and the brain initiates its nightly cleaning cycle, the aquaporin 4 channels, the specific cellular doorways that allow water to enter the brain, are fully supported, ensuring the swift and efficient evacuation of the neurotoxins that would otherwise spend the next decade quietly dismantling your memory centers. While sweeping the brain clear of pathological proteins via systematic hydration creates a pristine internal environment, this purified biological landscape must then be rigorously oxygenated and stimulated, leading us to the second, immensely powerful microhabit that marries physical movement with an incredibly specific respiratory technique. We have long understood that cardiovascular exercise is beneficial for the heart, but its profound impact on the aging brain is a relatively recent obsession within the field of cognitive neuroscience, primarily due to exercise's ability to stimulate the production of brain-derived neurotrophic factor, or BDNF. BDNF is often referred to as miracle grow for the brain, a potent protein that promotes the survival of existing neurons, encourages the growth and differentiation of new neurons and synapses, and is absolutely vital for long-term potentiation, which is the cellular mechanism underlying learning and memory formation in the hippocampus. Brisk walking, particularly when performed daily, drastically increases cerebral blood flow, causing shear stress against the endothelial walls of the blood vessels, which in turn triggers angiogenesis, the growth of new blood vessels, thereby expanding the vascular network that feeds the oxygen-hungry brain. However, simply putting one foot in front of the other is not enough to maximize this cognitive shield. The transformative magic occurs when brisk walking is strictly coupled with continuous, intentional nasal breathing, a physiological technique that radically alters the biochemical state of the entire body. Many adults unknowingly transition into mouth breathing when they exert themselves, but breathing through the mouth bypasses the intricate filtering and conditioning system of the nasal cavity, and most importantly, completely negates the production of a miraculous molecular messenger called nitric oxide. The paranasal sinuses are massive reservoirs of nitric oxide, a powerful vasodilator that is continuously produced by specific enzymes and then carried deep into the lungs only when we inhale through the nose. When this nitric oxide reaches the pulmonary alveoli, it massively expands the blood vessels, increasing oxygen uptake in the blood by up to 20% compared to mouth breathing, while simultaneously lowering blood pressure and reducing the workload on the aging heart. Furthermore, nasal breathing enforces a slower, deeper respiratory rate, engaging the diaphragm and triggering the parasympathetic nervous system, the rest and digest physiological state that actively suppresses the release of cortisol, the notorious stress hormone that has been clinically proven to physically shrink the hippocampus and accelerate memory loss when present in chronic, elevated levels. Beyond even these benefits, nasal breathing optimizes the Bohr effect, a physiological phenomenon wherein the slightly increased carbon dioxide levels retained by slower nasal exhalations actually prompt the hemoglobin in the red blood cells to release their oxygen molecules more readily into the surrounding brain tissue, ensuring that the critical memory centers are bathed in optimal levels of oxygen. Therefore, the daily microhabit of taking a brisk, 30 to 45-minute walk while keeping the mouth firmly closed and breathing exclusively through the nose acts as a dual-pronged neurointervention, simultaneously flooding the brain with BDNF through physical movement while utilizing nitric oxide and the Bohr effect to hyperoxygenate the newly expanded vascular networks, creating a robust, resilient brain architecture that is vastly more resistant to the decay of aging. This hyperoxygenated, rapidly clearing neural network must be continuously supplied with the precise molecular building blocks required to repair cellular damage and construct new synapses, a necessity that introduces the third critical microhabit revolving around targeted, anti-inflammatory nutritional interventions designed to protect the blood-brain barrier. The blood-brain barrier is a highly selective, semi-permeable border of endothelial cells that prevents pathogens, heavy metals, and circulating systemic inflammation from entering the central nervous system, effectively acting as the brain's exclusive security detail. As we age past 60, this barrier can become dangerously compromised, developing microscopic leaks that allow inflammatory cytokines from the body to infiltrate the brain, triggering a state of chronic neuroinflammation where the brain's resident immune cells, the microglia, become hyperactive and begin to attack healthy neural tissue in a misguided attempt to protect it. This slow-burning neural fire is now considered one of the primary drivers of all age-related cognitive decline, making the aggressive suppression of systemic inflammation through specific daily dietary habits an absolute non-negotiable requirement for cognitive longevity. The most vital component of this nutritional armor is the daily, calculated intake of omega-3 fatty acids, specifically docosahexaenoic acid, or DHA, which is literally the structural foundation of the brain, making up over 90% of the omega-3 fats found in the brain tissue and roughly 25% of the total fat content of the entire organ. DHA is seamlessly incorporated into the phospholipid bilayer of the neuronal cell membranes, ensuring that these membranes remain fluid, flexible, and highly responsive to neurotransmitter signaling, whereas a diet high in processed trans fats and excessive omega-6 results in rigid, inflamed cell membranes that severely inhibit synaptic communication and drastically slow down cognitive processing speed. Integrating a daily microhabit of consuming high-quality, cold-water fatty fish or supplementing with highly bioavailable microalgae oil ensures a steady stream of DHA to constantly rebuild the aging brain's physical structure. But this must be powerfully synergized with the daily consumption of neuro-protective polyphenol. Polyphenol are highly complex, deeply pigmented organic compounds found in massive concentrations in foods like wild blueberries, dark chocolate, green tea, and extra virgin olive oil, and they act as potent antioxidants that cross the blood-brain barrier to neutralize the free radicals generated by the brain's massive energy consumption. Furthermore, we must acknowledge the profound and undeniable connection between the gut microbiome and the brain, a bidirectional communication superhighway known as the gut-brain axis, where the trillions of bacteria residing in our colon actively produce massive quantities of essential neurotransmitters, including upwards of 90% of the body's serotonin, a neurochemical deeply involved in mood regulation and cognitive function. By establishing a microhabit of consuming diverse, fibrous, polyphenol-rich plant foods every single day, we actively feed the beneficial strains of gut bacteria, which in turn ferment these fibers into short-chain fatty acids like butyrate, a compound that systematically enters the bloodstream, travels to the brain, and directly strengthens the integrity of the blood-brain barrier, effectively sealing the leaks that cause neuroinflammation. This continuous daily ritual of marrying omega-3 fatty acids with polyphenol-rich plant fibers creates a fortified, radically anti-inflammatory neural environment where the microglia remain calm, the neuronal membranes remain fluid, and the sheer structural integrity of the brain is preserved against the onslaught of chronological aging. Even with a perfectly hydrated, oxygenated, and structurally sound brain, the physical hardware of the nervous system is meaningless without the continuous upgrading of its software, bringing us to the fourth essential microhabit, which is the daily enforcement of profound cognitive friction to build an impenetrable cognitive reserve. In the realm of neurobiology, there is a fundamental law first proposed by the neuropsychologist Donald Hebb, often summarized as neurons that fire together, wire together, a principle that perfectly encapsulates the concept of neuroplasticity, demonstrating that the brain's physical structure is dynamically altered by the experiences, thoughts, and challenges it processes. In our younger years, the demands of education, career building, and navigating complex social hierarchies naturally provide an overwhelming amount of cognitive friction, forcing the brain to constantly adapt, sprout new dendritic spines, and thicken the myelin sheath, the fatty insulatory layer that wraps around the axons of neurons and vastly increases the speed of electrical transmission. However, as individuals enter their 60s and often transition into retirement, their daily routines typically become highly predictable, comfortable, and automated, which represents an absolute biological disaster for the brain, because when a neural pathway is no longer challenged, the brain ruthlessly prunes those connections in an effort to conserve metabolic energy, leading to a rapid, measurable shrinkage in the brain's volume and a corresponding decline in memory capacity. To combat this, we must consciously construct a cognitive reserve, a surplus of robust, redundant neural networks that act as a massive buffer against age-related decline, allowing the brain to efficiently bypass any damaged areas by routing information through newly forged alternative pathways, a phenomenon that explains why some individuals with massive Alzheimer's pathology in their brains never actually exhibit symptoms of dementia during their lifetimes. The microhabit required to build this reserve is not the passive consumption of entertainment, nor is it the mindless repetition of simple crossword puzzles, but rather the daily, uncomfortable engagement in a state of active, highly demanding learning that pushes the individual slightly beyond their current intellectual capacity. This requires dedicating at least 30 focused minutes a day to a completely novel and complex task, such as learning a new foreign language, which fundamentally alters the gray matter density in the bilingual cortex, or learning to play a musical instrument, which requires a profound, simultaneous integration of the visual, auditory, and motor cortices, forging incredibly strong cross-hemispheric connections via the corpus callosum. Even the act of utilizing your non-dominant hand for daily tasks like brushing your teeth or operating a computer mouse forces the motor cortex to work outside of its deeply ingrained, automated patterns, triggering a mild state of beneficial neurological stress that signals the brain to release nerve growth factors and reinforce its synaptic architecture. By purposely injecting this intense, deliberate cognitive friction into the daily routine, the aging brain is essentially tricked into believing that it is still in a phase of vital, early life development, refusing to shut down its neurogenic machinery and instead continually weaving a complex, unbreakable web of neurons that ensures memory retrieval remains lightning fast, problem-solving remains sharp, and the capacity for intellectual growth remains totally unhindered by the passage of decades. All of these meticulously crafted microhabits, the strategic hydration flushing the glymphatic system, the hyperoxygenation of nasal breathing during exercise, the structural reinforcement of anti-inflammatory nutrition, and the intense synaptic building of cognitive friction, ultimately converge and rely entirely upon the execution of the fifth and final habit, which involves the absolute biological mastery of sleep architecture and the optimization of the circadian rhythm. It is a dangerous and widely held misconception that the elderly naturally require less sleep than younger adults. The reality is that the aging brain desperately needs the exact same 7 to 8 hours of restorative slumber, but profound changes in the body's circadian timing system make achieving this deep sleep significantly more difficult without intentional, rigorous behavioral interventions. The circadian rhythm is the internal 24-hour biological clock, governed by a tiny cluster of thousands of neurons located in the hypothalamus, known as the suprachiasmatic nucleus, which relies primarily on environmental light cues to regulate the massive daily fluctuations in hormones, body temperature, and cellular metabolism. As we age, the photoreceptors in our eyes naturally degrade, and the lens yellows, significantly reducing the amount of light that reaches the retina. which in turn causes the suprachiasmatic nucleus to receive weaker signals, leading to a fragmented, confused circadian rhythm where the brain struggles to differentiate between day and night, drastically suppressing the nightly release of melatonin, the master hormone of sleep. Furthermore, this circadian fragmentation severely curtails the amount of time the aging brain spends in non-rapid eye movement slow-wave sleep, the deepest phase of rest characterized by sweeping, synchronized delta brainwaves, which is the exact period when the hippocampus acts as a rapid replay mechanism, transferring the fragile, short-term memories of the day into the stable, long-term storage vaults of the prefrontal cortex for permanent consolidation. Without adequate slow-wave sleep, this transfer completely fails, and the memories of the preceding day are simply overwritten and lost forever. A terrifying phenomenon that perfectly mirrors the rapid memory decay seen in the early stages of dementia. To reverse this sleep degradation, the final essential microhabit requires anchoring the circadian rhythm with intense precision, starting with the immediate exposure of the unprotected eyes to natural sunlight within the first 30 minutes of waking, which sends a massive, undeniable signal to the suprachiasmatic nucleus to halt all residual melatonin production, and begin a chemical countdown that will precisely trigger a surge of sleepy hormones roughly 14 hours later. This morning light viewing must be paired with extreme temperature regulation in the evening, as the brain must drop its core temperature by roughly 2 to 3 degrees Fahrenheit to even initiate the biological cascade of slow-wave sleep, meaning that the bedroom environment must be kept aggressively cool, ideally between 60 and 65 degrees. By avoiding blue light-emitting screens in the hours leading up to bed, which falsely signal to the brain that the sun is still shining, and perhaps employing a hot shower before sleep to draw the body's heat away from the core and into the extremities for rapid cooling, the individual systematically overrides the aging defects of the circadian clock, forcing the brain into deep, uninterrupted cycles of slow-wave and REM sleep. It is within this profound darkness and perfectly regulated temperature that the magic of all the previous habits truly manifests. The brain literally shrinks to allow the glymphatic waters to wash away the day's toxic amyloid proteins. The newly oxygenated and nutritionally fortified synapses solidify their structures, and the immense cognitive friction of the day is smoothly converted into permanent, resilient intelligence, completing a continuous 24-hour cycle of relentless neural preservation. When viewed as an isolated task, the prevention of cognitive decline after the age of 60 can appear as an impossibly daunting mountain to climb, a terrifying battle against the inevitable forces of biological decay. But when this massive undertaking is deconstructed into these 5 deeply scientific, highly manageable microhabits, the preservation of the brain transforms from a desperately hoped-for miracle into a highly predictable, biologically guaranteed outcome. We are not simply victims of our genetics or passive passengers on the ride of aging. We are the active architects of our neural destiny, possessing the incredible capacity to physically mold the microscopic structure of our brains, right up until the very end of our lives. By meticulously flushing out the microscopic debris through timed, precise hydration, we refuse to let the neurotoxins build their suffocating plaques. By insisting on closing our mouths and forcing the air through our nasal passages while we rapidly move our bodies, we unlock ancient biochemical pathways that flood our nervous system with life-giving oxygen and neurotrophic growth factors, essentially breathing youth back into our decaying vascular networks. By treating our daily meals not merely as a source of pleasure or basic caloric energy, but as highly targeted pharmacological interventions rich in brain-building lipids and inflammation-quenching plant compounds, we provide the raw, structural materials necessary to rebuild the incredibly complex walls of our cellular machinery. By courageously stepping outside of the comfortable, predictable routines of retirement and relentlessly subjecting our minds to the beautiful struggle of difficult, complex learning, we weave a cognitive safety net so thick and resilient that the physical pathologies of aging simply cannot break through to steal our memories. And finally, by bowing to the ancient astronomical rhythms of light and dark, fiercely protecting our sleep architecture, and allowing our brains the sacred, silent time necessary to consolidate, clean, and repair, we ensure that every single effort we make during the waking hours is permanently forged into the biological reality of our minds. This incredibly intricate, seamless integration of habit, biology, psychology, and neuroscience provides an absolute masterclass in human longevity, offering far more than just the mere extension of life. It promises the preservation of the self, ensuring that those precious, hard-won years after 60 are not defined by confusion, loss, and dependence, but are instead profoundly characterized by enduring clarity, boundless resilience, and a brilliant, unyielding vitality that allows us to experience the culmination of our lives with the absolute fullness of our consciousness intact.